Okay. Awesome. Well, here we are, mycology class. There are apparently 18 students. We've got half of you, less than half of you actually here right now. So hopefully people will be joining us shortly as we get this figured out. Any questions before I get started? What sort of things are you deeply concerned about as we get the semester rolling?
Not deeply concerned, but I was wondering, you want us to get that microscope thing off of.
Yeah.
What are we going to be using that for? Are we going to be buying some slides to look at, or?
I've got a little packet I'll be giving you. I'll go into detail about that, but this thing will actually let you like find fungi for free out in the real world and look at it, and it hooks up to your cell phone. So,
Okay, cool.
Yeah, I'm justifying this. I know it's $20 you didn't expect to spend, but I'm not making you buy the textbook. So I have a stolen version of the textbook I'm going to send you.
And if we have another microscope, you still recommend buying this one? I mean,
No, if you have a microscope already, that's great.
Yeah. Okay, awesome.
Yeah. This is just the cheapest microscope that actually works. Right. Yeah. Okay.
And are you going to send a link to where we can find one of those?
Yeah. Yeah.
Right.
I'll walk you through that today.
Okay.
All right. So I guess let's go ahead and get started. I'm going to share my screen with you. All right. Let me know if you can see that.
Yep.
All right. So our course has its own little web page that's linked in Canvas, and I believe I sent it out to all of you as well. Use this kind of like our roadmap. It's kind of like a living syllabus where you can actually click on the links to assignments and all that kind of stuff. So it's got a link to the actual syllabus, so let's just go through that real quick. Okay. There's our website. There's our textbook. Don't buy it. I'm going to give it to you. Okay. Learning objectives, and then probably what you're most interested in is kind of this course structure and grading. Typically, I teach this class as a course-based research experience where all semester long you are working with a group on a research project involving fungi. That is not going to be possible this semester, so I apologize. So I've gotten rid of those points and just thrown them up here into making basically your quizzes and discussion worth more. Okay. Here's our grading scale. A lot of what's going to happen in this class is I'm going to have lecture materials. You'll have access to all those slides. I'm recording all of our meetings so you can have those to study from. There'll be outside reading, usually maybe about 8 to 10 pages worth from our textbook, and then a shortish scientific paper. And we're going to have Thursdays be kind of discussion focused and Tuesdays be more lecture and lesson focused. Okay. We're also going to have lab assignments, and that's kind of where the microscope that you have or buy will come in handy. People think of fungi and they think of what?
Mushrooms.
Mushrooms. Yeah. Right, mushrooms. And there are a lot of different mushrooms out there, but the diversity of fungi tends to be pretty microscopic, and so it's a microscope-based class, pretty much. All right. We'll have 3 exams, and that includes the final, 100 points apiece. They are open note, open internet, open lectures, videos, slides, all that kind of stuff. Okay. Any questions about anything so far? Nothing?
Are the exams going to be timed?
It's going to be like 24 hours. Yeah.
Oh, okay.
Yeah.
So you said
I'm trying to be pretty generous and lenient with regard to that kind of stuff. We're all going through. Oh. Am I back?
Yeah.
Okay. Yeah, see, this kind of crap is obviously going to happen. Internet goes out or whatever. So that's why I'm given 24 hours for exams, and they're all open based on our readings and lecture materials. Okay.
So Dr. Zahn, you said so the tests are like open book, open note, open internet.
Uh-huh.
So some of the other classes they're taking the test through like this closed browser type thing.
I don't believe in that.
Okay, cool.
Yeah.
Just wondering.
To me, that feels really invasive, especially when they have the proctoring people, like some person
Staring
in
at
India
you.
staring into your room. Like, that's creepy.
Yeah. Okay.
Yeah, no.
Sounds good. We like you. So yeah.
Great. Perfect. All right. And then the rest of the syllabus is the boilerplate policies and things. That's why syllabi are now 40 pages long. Okay. So that's there for you. This link right here, can you see that?
Yeah.
All right. You click on that and it's going to give you a PDF. That's your textbook right there that we'll be using. So feel free to download that. Don't try to sell it to anybody or you might get hit with like copyright violations or whatever. Don't tell on me that I'm giving this away for free. Okay. So that's our textbook. Keep that handy. And then for the lab, there's actually quite a bit you can do with mycology at home without having access to like DNA sequencing and stuff. Ideally, normal semesters, we'd be doing some DNA analysis of fungi, but I call this hillbilly mycology, and it's how I actually got started in mycology, was doing stuff at home. All right, so equipment, the only thing you need to purchase for this class is the microscope. And so click on that. It's the Amazon link. Last time I checked, it was 20 bucks. Oh, down to $18. All right. So go ahead and order that fairly soon so we can have it for, you know, next week or two, doing some stuff with our labs. This page is just kind of, I started putting it together as a resource for other mycologists that were teaching online and had questions about what kind of stuff we can do with labs at home. So I just started kind of collecting stuff from the internet or other people's helpful resources and sticking them all in this one spot. So it's got a lot of resources for how to do things at home really cheap with mycology. For our labs, the only thing you got to buy is that microscope or have a microscope that you can use. So if you are on campus a lot and in a classroom with a microscope and you want to do your lab there, that's fine. Okay. What was I saying? Oh yeah, that's the only thing you got to buy. The rest of the stuff, I will have a little packet for you. Okay, that's almost done right now. It should be finished by tomorrow. And I'm going to stick that in the biology main office so you can come and pick it up. If you can't make it there, please let me know and we'll find a way to get it to you. What's in there are, you know, some slides and cover slips, some fungal cultures. You're going to get a whole stack of petri dishes that we can use to isolate fungi from different sources. A little tube that looks like it's empty, but it's not, so don't open it. It's got a little bit of pine pollen, and we can use that to catch chytrid fungi. So other things you might need are probably things you have around the house already, like tweezers, right? Or just like a little kitchen knife to act as a scalpel, that kind of thing. So it should be pretty straightforward, and once you get your kits, we'll talk more about the upcoming labs. Okay. So that's our quick links right there. So if you're looking for a protocol, that can link you to videos about how to isolate zygomycetes. Okay, so these are just some that I'm demonstrating the use of this little microscope with some moldy bread, essentially. All right. We're going to isolate chytrids from whatever water source you want. We'll do some slide culturing. Hopefully be able to find some insect-associated fungi as well. One fun thing is to catch a fly and let it just walk around on the auger, and you can get all the fungi that live on its feet. And there'll be bonus points if you manage to grow mushrooms at home. Okay, so I'm going to show you how to do that. You'll have the resources for it. All right. Questions about hillbilly mycology before I move on to the rest of the website?
Could you repeat that last paragraph? Sorry, you were going in and out, breaking up a little bit.
One of the projects that I had planned, but it's maybe not going to work for everybody because it just doesn't always work, is growing mushrooms, like edible mushrooms at home. And so we're going to walk through that and try to get it to work. And if it works for you, great. If it doesn't, also great. But at least you'll know how to grow mushrooms. And so that's kind of like a bonus lab assignment that we'll get to towards the middle of the course.
And bonus dinner if it works.
Yeah, when it works, it's great. You get like free oyster mushrooms. All right. All right, so I got some other web resources for you. This is another like online textbook, and this is just a download link. It's kind of an older edition, but it's free. It's got a lot of nice stuff in it. Optional. It might be really useful for this class, or if you're stuck on a test question or something, it may have an answer. Some other websites kind of based around different aspects of mycology. Okay. The fungal gene database. Obviously, we're not going to actually get to do any DNA sequencing this semester, but we can use DNA sequences that other people have been through because I want you to know how to analyze the DNA. So we'll walk through that. Okay, and this brings us to the meat and potatoes of the website. I've divided it up into different weeks. And so we've got our main topics, your readings and assignments for the week, and then resources. Okay. The lesson slides are already there, but the video, obviously, we're recording it now, so I will upload a link there after class is over. So in case you have to miss class or something, you'll still have easy access to what went on. Okay. And then just like little videos that help walk you through what we're talking about in lecture. Okay. Questions?
Hey, Dr. Zahn, I got a question for you.
Great.
Where on Canvas is the link to the website?
Announcements, I believe. Maybe. Yep. It's the first announcement.
Okay. Thank you.
Mm-hmm. Yeah, go ahead and bookmark this website. Use it like a roadmap for the semester. Each week, it'll have all your readings in one spot, all your assignments listed, that kind of stuff. The way this class will typically work, I mentioned this briefly before, is that we're going to have lesson time on Tuesdays and then quizzes on Thursdays. And those quizzes are going to be on Canvas and they're due before class. Okay. So you can start your reading now. You don't have to wait until after the lesson's over. The reading typically isn't a ton. It may take you an hour. I don't think there are any reading assignments that would possibly go over an hour unless you're a really slow reader. So schedule for that. Okay. The quizzes are also open book, open reading. So, I mean, I wouldn't go into it without having done the reading, thinking that you're just going to find all the answers in a reasonable amount of time. That's not going to work out for you. Do the readings, take the quiz. All the questions are going to be visible to you, and you can use the reading to help fill in gaps of your memory. That sound all right?
Sounds great.
Okay.
So did you say Tuesdays are lecture days, and then Thursdays is during that time we're taking quizzes?
The quiz is due before class on Thursday.
Gotcha. Okay.
Yeah.
Sounds good.
And then during class on Thursday, we're going to be working on like protocols for lab stuff and doing discussions for the readings. All right. And you do get participation points for being there and asking questions and talking about the papers that we read. Okay. So I guess this is a long way of saying if there's a day of the week you're going to have to miss, make it a Tuesday rather than a Thursday, because you'll have the video and you can come watch the lesson, right? And you won't miss any participation points that way. Okay. So that's kind of the structure of the class. Coming up, where's our first exam is in week 5, where we cover the material from weeks 1 and 4. We'll have 24 hours to take that exam, open notes and everything. But the class is basically broken down into a few sections, and I think I put them up here. Yeah. First up, we're just going to kind of go through the phylogeny of fungi and learn about the major groups. Okay. This will seem like it goes kind of fast. You know, in 4 weeks, we're covering all of the fungi almost, and there's going to be a lot of jargon and terminology thrown at you. Okay. Reading is going to help with familiarity of those terms. So if I say basidiospore, I expect you to know what that means after we cover basidiomycetes. Okay. Then we're going to go look at fungi as the enemy, right? So the pathogenic fungi and how they get around the world to infect people or crops or stuff like that, and the kind of weird poisons and secondary compounds they make. And then my favorite, the fungi as a friend, where we talk about symbioses and mutualisms. Okay. And then towards the end of the semester, we'll go through molecular mycology. And so, like I mentioned, we're not going to do any sequencing, but we are going to have fungal sequences, and I'm going to hopefully get you to the point where you'll be able to kind of analyze those on your own using online resources without any coding knowledge. Okay. If you get down towards the bottom of the website here, you'll see that it's kind of sparse, and that's because I'm not quite done building it yet. I'm still adding resources and lessons for those sections, but the first 5 weeks are pretty well filled. So those will kind of pop up as I get to them. Okay. So your first reading assignment for this week is James et al. 2006. It's a bit of an older paper, but this one was really groundbreaking. Okay. It made it into Nature, which is a really prestigious scientific journal. And look at that list of authors. So dozens of people spent a lot of time basically to make this phylogeny. All right. And they were really trying to get at this divergence of fungi and animals, because fungi and animals are each other's closest relatives. Right? Fungi are way more similar to animals than they are to plants. And so they're trying to look at the first fungi and that early diverging group. We know them as the chytrids or the chytridiomycetes. Okay, and so that's what this paper is about. And so it's a good place to start our reading. All right. So make sure you read that. Keep in mind what the authors are trying to accomplish and how they did it, and that'll help you on your quiz. And then for any reading that says Webster and Weber, this is giving you the chapter and section numbers, and that's our course textbook here. So for the first bit, Webster and Weber 1.1, 1.5, and 6.1. Those are just listed here on the side if you have it in chapter view. And so that's what are the fungi, the taxonomy of fungi, just this section. Okay. And then 6.1 the introduction to our first group of fungi, the Chytridiomycota. Okay. So that's your reading that your quiz will be about. You've got a video to watch, and then I want you to start finding and growing zygomycete fungi, because it takes a couple weeks, so it's a good time to start right now. All right. So that assignment is up on Canvas already, and we've got a video walkthrough right there for you. What you will need is a Ziploc bag, some dirt, and a slice of bread. Really not hard. We're making moldy bread. That's your assignment. And then make sure you have access to a microscope, or order this one if you want a nice cheap pocket microscope. All right. Are we ready to start lesson 1?
Yeah.
I got 1 vote for yes.
Sure.
Rock on. Do it.
2. I'll open the little meeting chat here. If you have anything you want to say, throw it in there. Okay. But I can click on slides, so you'll have access to these. Okay. I'm just gonna open her up. Hopefully any second now. Yes. Okay. So good spot to start is with the beginning of fungi, and so we want to think about what fungi are and when and where they evolved and what makes them different from the most recent common ancestor of fungi and animals. So we're going to look at their evolutionary history, the types of growth habits that we find in the kingdom fungi, some physiology, a little bit about their diversity, and then really finish up with the chytrids. Okay. So you may see these called Chytridiomycota or the Chytridiomycetes, or the slang for it is just chytrid. Right. So be familiar with that. That's one of our main 5 groups of fungi that we're going to be covering here. And they're really weird because they're quote unquote primitive fungi. They still retain a lot of the characteristics of the common ancestor that they had with animals. So on this picture here, this phylogeny, imagine that down here at my cursor is the common ancestor of fungi and animals. What's this look like? Labeled number 1 here.
Looks like a simple bacteria with a flagella.
Okay, any other thoughts? I hope it's not bacteria, because we're talking about common ancestor of all fungi, which are eukaryotes. It looks like a sperm to me. All right, and that's basically what it is. Animals all have that same structure, right? That's animal sperm. But the earliest fungi also had that, and some of them still retain it to this day. This little single-celled, flagellated, with 1 flagellum that kind of pushes them along, right? So we're talking about the eukaryote supergroup Opisthecont, if you remember that from earlier biology classes, and it means single pushing flagellum. All right, so the chytrids are the fungi that still retain that single flagellum. All right, so it's showing it there. And then here's our chytrid groups. We're not super sure about their deep evolutionary relationships because doing genomics on them has proved to be kind of difficult, and they're kind of hard to find. Some of them only live in the guts of cows and deer. So this is kind of a tough group to study. Here we see the evolution of multicellularity in fungi, labeled number 2. Okay, and so that's our zygomycetes and things, and then we've got our fungi that we call mushrooms. Okay, so the basidiomycetes. But what we're really talking about right now is kind of this early diverging section. All right, so what are fungi? Other than what I've written on this slide, what are your opinions? Either write them in the chat or yell them at me. What defines a fungus?
Period that has a cell wall, usually made of chitin.
Yep, okay. Cell wall with chitin. And that's almost always true.
Almost.
Yeah. In fact, mycology is a tough thing because fungi break all kinds of rules, even their own rules. And some of the things that we do consider fungi actually have cellulose cell walls. They may not be considered fungi in a year or two. Who knows? I actually don't care. But let's go with chitin cell walls as a pretty good determinant of fungi. All right, what else? What have you heard?
They use spores to reproduce.
Use spores to reproduce, okay. I'm trying to get a feel for what all of you think about fungi, what your preconceptions are. Is everyone shy? You afraid of having the wrong answer?
Well, obviously, when I think of fungi, I think of mushrooms and bread mold and bathroom mold. That's just instantly what first comes to mind.
Yeah, and those are obviously the most famous fungi, right? The stuff that ruins your sandwich, makes your bathroom gross, or tastes yummy on a salad or pizza. Yeah.
If
But
they get their food from other sources.
yeah, so they're heterotrophs, definitely. Jonathan has written decomposers. Some fungi, actually, the vast majority of fungi are decomposers, but I wouldn't call it a hard and fast rule. Yeah. All right. Well,
Non-motile, or whatever, however you say that word. They don't move usually.
usually, well, they don't move. I think I'm going to say no on that one for a few reasons, and we'll see why here in a second when we start looking at chytrids. But I mean, look at that.
It looks motile, yeah.
Yeah, it's totally motile. And fungi definitely get around on your sandwich. They have little hyphae that move around, so
Right.
they're
Yeah.
definitely slower at moving than animals, but they're pretty motile. Okay. They diverged from metazoans, the animal group, long time ago, estimated about 1 billion years from our earliest fossil records. This is kind of in dispute, but it's safe to say at least 800 million years ago. Okay. So they've had a long, long time to diverge from animal groups, and yet they still retain a lot of the same biochemistry. They're still heterotrophs. Some of them still have that sperm-like structure. Okay. These are fossil chytrids pictured here. Okay. And we just found these last year at 1 billion years ago. Some really, really old rocks in southern Australia. And those are like most definitely chytrids. So I'm of the opinion that this 1,000 million years is a pretty solid answer based on these fossils alone, unless they age the rocks wrong. Okay. So they've had a long time to diverge from animals. It's really tough to define what fungi are without just saying, oh, it's this monophyletic group. Okay. It's kind of a circular argument for what fungi are. But when you look at this is opisthecons here. So we've got animals and choanoflagellates, and then the ichthyophores, and then here's all of our kind of fungal things. We're looking at this most recent common ancestor. You'll see this abbreviated as MRCA a lot in papers and in our textbook. So get used to that acronym. It's just referring to the closest relative, hypothetical relative of any two groups. And so we're saying the MRCA of fungi and animals is this little point right here. Okay. What does that have in common with what we see now in animals and fungi? That's one of the kind of questions that evolutionary biologists try to answer, to try to track back where all the changes happened. Right? So how far back did they have certain traits? Well, we see the flagellum in some of these. These have been considered to have a newly acquired flagellum or more recently acquired flagellum, but the common ancestor did have a flagellum. It was a sexual species. It had something resembling an alternation of generations, okay? And they both had chitin. And so I think it was you, Christina, who pointed out that fungi have chitin cell walls, right? What animal groups have chitin?
Insects usually.
Yeah, insects, all the arthropods.
Right.
So the shell on your shrimp, that's chitin, okay? And so it kind of makes sense that the most recent common ancestor had that as well, and we see a lot of good evidence for that, okay? This particular phylogeny right here, excuse me, it's showing some of the major morphological changes, all right? And so these little red things with the flagellum crossed out, it's saying that the flagellum was lost in these groups. And in the green, it's saying a new flagellum was gained in those groups, okay? And we've got osmotrophy that's only found in the ichthyophores and the fungi, not in animals. And so we're starting to kind of notice some differences between these groups, okay? So this class, mycology, is in some ways looking for differences between fungi and animals and trying to exploit those to treat fungi more reasonably. In the past, what did everyone think fungi were phylogenetically? Plants, right? They were considered just weird plants, and everyone thought that they were plants. They grow like a plant, you know, up out of your lawn after you mow it. They produce spores just like a fern does, and so the botany departments around the world got filled with mycologists where they don't belong, right? It'd actually be better to stick them in with a microbiology department or with like a zoology department because they're more closely related with animals, okay? Historically, when we look at the fossil record, we see that fungi made it to land the same time as the earliest algae, okay? So when was that? Anyone? I'll give you a second to look at this figure. I'm going to blow it up for you.
Is it around that 300 mark?
Earlier.
A little earlier than that.
So 300, we're looking at expansion of dryland forests already. So we got like vascular plants taken over.
Around 420?
Yeah,
Or so.
about 425 million years ago. And, you know, possibly earlier than that. Okay, so possibly up to 500 million years ago, some of the really first trace evidence we see of like algal fossils and biochemistry in some of the rocks there on land. And that's when we start seeing fungi as well. So right in through here, okay? So fungi are showing up on land along with some goopy algae, all right? Way long time ago. But I told you that fungi diverged from animals how long ago?
$800 million.
Yeah, 800 million to a billion years ago. And so fungi are already kind of their own thing. They've had a long time 500 million years, roughly, of evolution since they split from metazoans. Okay, so we've got fungi, they're their own thing, and they're showing up on land with the first algae, and they're already interacting with that algae. Some of the first things we see are plant cells, like little simple algae, in super close association with fungal cells. All right, and we can even do the biochemistry on those fossils and see that the plant has cellulose and the fungi has chitin at that point, and they're interacting really closely. So the thinking here is that algae and plants couldn't have actually arrived and survived on land without fungi, because we don't see examples in the fossil record of early algae or mosses that aren't associated with fungi. Okay, and when we look around today, like 80% of all plants in existence depend on fungi. All right, so the thinking here is that you can actually define plants as algae that figured out how to live with fungi really well. Okay, so fungi are showing up on land real early. That's nice for the plants because it helps build soil, it helps recycle nutrients for them, it helps them get access to things like nitrogen that may have been in pretty short supply on land. Okay. Here's some pretty early fungal fossils. I mean, artist's depiction of them in the top right, the guy standing next to one here in this photo, and then the bottom photo is an actual fossil of a fungus. So this is about 400 million years ago. This blows my mind. I'm sorry. This is crazy town for me. I wish we still had fungi like this around. Okay, but for a while they were the dominant organisms on land in terms of size. Okay, why might that have been the case? And why aren't they the dominant large organisms on land now?
I would think that it's multiple factors playing into it, such as air, the combination of the molecules within the air, as well as different species feeding on them.
Okay, so animals were starting to show up at this point, but they were things like roly-polies, you know, millipedes and stuff. So that's basically the only animals. Those still eat fungi to this day, and so they probably had some predators around, but they were pretty small. What do you not see in this artist's rendition of the landscape?
No plants.
I see some green stuff.
Oh, I guess, yeah. Very little water.
Yeah, I don't see any water, but it's pretty hazy here. I mean, this is an artist rendition, obviously, but it was raining. The early plants that showed up, these are not vascular plants. You know, they're like mosses and that kind of thing. Liverworts, little patches of algae. The air was different. It had higher CO2 at this point because there weren't giant trees anywhere, right? So vascular tissue hadn't evolved yet. And until vascular tissue evolves, these early plants can't suck up a ton of CO2 from the atmosphere, or when they do, they quickly die and decompose. So there's no trees taking over, there's tons of CO2 in the air, so these plants are doing pretty well, but they fall over and there's nothing to decompose them because nothing can decompose cellulose except for fungi. And so fungi became the dominant critters on land for quite a while there because there were just so many dead plants everywhere, right? It's a massive food source. There weren't tons of animals running around eating them or eating the plants and running away with them, that kind of thing. So we get these giant fungi that dominate the landscape for about 50 million years. Okay. Fast forward to today, and we don't have, I'm sorry to say, those gigantic fungi anymore, right? We've got things like bread mold, baker's yeast, okay? These are the main growth habits of fungi. We got 3. Mycelial, this is by far the most common, and that's the little threads, right? Looks like spaghetti noodles going through things, okay? That term mycelial, get used to it, all right? Those little filaments are called hyphae. That's plural, hypha is singular, okay? Then you've got the yeast growth, and that's single-celled, right? So it's just little blobs. That's what baker's yeast looks like, and you can see it kind of budding off here, right? Then you've got dimorphic growth, which means they just do both the mycelial and the yeast growth form, okay? Most of our medically important fungi are this dimorphic growth form, where they like to spend their lives in this single-celled state, but given the right environmental cues, they'll move to a hyphal mycelial growth form and kind of start eating your flesh or your fingernails or whatever. Okay. So, name the fungal growth habit. I'm going to start at the top with this little girl. Anyone?
Is that one called a fairy ring?
It is a fairy ring, but is it mycelial, yeast, or dimorphic? Don't be afraid, people.
I would guess dimorphic.
Okay, we got one for dimorphic. What makes you think that?
Just because of like the large part on top, but I know like mushrooms kind of have these super like little kind of hairy roots sometimes.
Yeah, all right. So those hairy roots that you're talking about, those are actually mycelia or hyphae. That's the, yeah, like the filamentous growth. This fairy ring here actually began right in the middle of it. And it started growing out in a circular pattern with those hyphae. The mushrooms that you see are just essentially the fruit. Okay. The main body of the fungus is actually underground here in this mycelial form, this filamentous form. It's going all through the soil there. I bet there are 40,000 miles worth of mycelia in this little patch. And the mushrooms themselves are just the sexual fruiting bodies. So when it starts running out of food or it turns cold out, like winter's coming, mushrooms will start to sprout out. And that's because the growth season's almost done. It's ready to have sex, do some recombination, and send its spores out. Okay. So this one's actually mycelial. It's filamentous. What about the toe?
Yeast.
Sometimes a yeast.
So it'll be dimorphic then.
Dimorphic, yeah. So if it's sometimes a yeast and sometimes filamentous, it's dimorphic. The giveaway here is that this is a human fungal disease, and most of those are dimorphic fungi, right? So it's kind of a safe guess. With that in mind, what about the tongue?
But would that be the same dimorphic?
Dimorphic, yep. Awesome. This one here is yeast, it's single-celled, and the rest are actually filamentous. Okay. Most fungi are actually filamentous. There are relatively few dimorphic ones and relatively few yeast forms. The yeast fungi are pretty cool. We obviously appreciate them because they give us bread and stuff, but they're really boring to look at to me. They all look exactly the same. Okay. So, you guys told me that the cell wall is made of chitin in fungi. What is chitin?
Carbohydrate.
It is a carbohydrate. How is it different from cellulose?
The arrangement. It is more. It's not parallel. It's so basically it's a harder carbohydrate than cellulose.
Yeah, it kind of does this back and forth linking, and it's got an extra like amine group. It's got nitrogen stuck in there also. Okay, so most fungi are filamentous like this. All fungi, except a few that might not actually be fungi, have this chitin cell wall. Okay, so here's our fungal cell wall. There's our chitin cross-linked together. Another major difference that fungi have is that they use ergosterol instead of cholesterol. Okay. They have absorptive nutrition intake, and they make a ton of secondary compounds, way more than plants or animals combined. Right. So we'll get to look closer at kind of fungal cellular structure here in a little while. I don't want you to get too hung up on it. There's a million things you could learn about fungal ultrastructure beyond this class. Okay. But chitin is essentially cellulose. Right? At the bottom here, this is cellulose polymer. Chitosan is found in some groups, and there's chitin. So can you see the difference? Instead of this OH, it's got this acetylamine group here. All right, so we replace the hydroxyl group with that, and that's basically the main difference between cellulose and chitin. So it's not super huge, and there are plenty of groups around the eukaryote tree of life that can make chitin. Okay.
Quick question.
Yes.
Is chitin stronger than cellulose?
It's kind of like an analog to keratin in animals, so kind of like your fingernail. It's a bit more flexible, and so in that sense, it's kind of harder to break than cellulose, but cellulose is more rigid, if that answers for you.
Yeah, that's fine.
Yeah, I wouldn't say that it's stronger in all situations. I mean, think about like a crab shell can be pretty tough. That's chitin. But it also flexes better than cellulose, so it's more resistant to like sudden pressures. Yeah. All right. So we've got a chitin cell wall formed around these mycelial shapes, and you told me that fungi are non-modal. I'm going to tell you that they actually are modal, and here's them growing. Okay. This is kind of tough, but they use water pressure to grow, and they digest themselves from the tip. It's really odd, right? So just like this guy blowing up balloons, fungi grow from the tips based on pressure behind them. So this is looking at the tip of a fungal hypha right here, all right? And you can see it's got its chitin stuff forming its cell wall. As you pointed out, chitin is pretty tough material, and so how do you expand that with just water pressure? The answer is all this crap that's going on in the very tips, okay? They in fact get a special name called the Spitzenkörper. Yeah, say that 3 times, all right? Spitzenkörper is a really active region of growth at the tip of every fungal hypha out there, okay? And it starts dissolving itself just at the tip. It actually makes chitinases, which is kind of weird. So it makes an enzyme that dissolves itself. It excretes it in the tip there, and the water pressure behind pushes the fungus forward. Right? So that's how these things grow. We'll learn more about them future lecture. Because now we need to talk about fungal nomenclature. Any questions up to this point about like the shapes of fungi, their evolution or the fact that they're motile and they grow through surfaces? Nothing? Oh, I see. How was the fungi fossilized? I have no idea. Sorry. Same way anything is fossilized. Gradual replacement with minerals. In that time on Earth, there weren't many decomposers other than fungi. I mean, there are bacteria, but they can't break down chitin very well. And so you probably have this fungus falling over, getting buried in an anaerobic environment. And so it'd be pretty tough to break down. Think of like the peat bogs in like Scotland, okay, where you find like mummified human remains that are still basically intact from 2,000 years ago. That's my expectation. Any other questions?
I have a question. With the growth, the tip, do they shed? You said that they shed. They basically digest themselves. Do they shed like the outer layer, or it's from the inside out?
From the inside out, they're
wow.
digesting their own cell wall. Yeah.
Oh.
They've actually made mutants of fungi to see if they could like get them to kill themselves, and they can. So the enzymes they're making are digesting their own cell wall from the inside out. And a fungus that's doing a good job is going to digest it just enough to make it flexible enough to expand, but not too much to where it bursts. Yeah. All right. As we get on in our lessons, especially during the first like fourth of this class, you're going to hit tons of brand new nomenclature. So let's take a look at that. Fungal nomenclature is really stupid. I'm sorry about that. Okay. This is due to historic reasons because people used to think they were plants. So the International Code of Nomenclature for algae, fungi, and plants governs how things are named, what they're called, and all that. And it's, of course, based around plant traits, not fungal traits, right? So even the seal here of this International Code of Nomenclature doesn't even include fungi in it. They're so unimportant, right? I don't know how to get around this, but currently we're stuck with plant rules for fungal names, right? Myco is something that you'll see really often. That just means fungal. EU means true, and it usually means a group that's monophyletic. So the Eumycetes means the true monophyletic group of fungi. Okay. If you see a word ending with mycota, that means a phylum or division. So Ascomycota is the Ascomycete phylum. Chytridiomycota is the Chytrid phylum. Right? Mycotina is subdivisions, so you sometimes see that, especially in your textbook, you'll definitely see Mycotina. Mycetes means classes, and Mycetales is order. All right, so Agaricomycotina is a big group of fungi within the Mycetes. Okay. Eumycota means all of the true fungi. So keep this in mind. When you get to your textbook, you're going to see these terms, and they're going to be overwhelming at first. So just remind yourself that these refer to levels of classification. Okay. Because we're stuck with plant naming rules for fungi, it doesn't always work, and nobody cares enough to fix it for real, or at least the plant people say, no, you have to keep naming them according to our rules. Sometimes the taxonomy breaks, all right? So we're going to look at this closer later when we start looking at the benefits of sex. But fungi often have two names or even more. The same species will have the same or two different names. This is a good example right here. We've got Nectria and Fusarium. That's the same fungus. This is just the asexual form, Fusarium is, and Nectria is the sexual form. Okay, we call these teleomorph and anamorph. It's easy to remember. Anamorph, asexual. Okay, so anamorphs are asexual, teleomorphs are sexual. This wouldn't have been a problem if we had started doing genetics long time ago and before we started naming fungi, but that wasn't really a thing because we started naming them in the 1700s. So we looked at fungi with the earliest microscopes, and we started finding these structures here, and anything that looked like that got the name Fusarium. Okay. These have different looking structures, so they got to be their own genus, Nectria, and we didn't put it together for a couple hundred years that these were the same species, just different stages of their life. All right. This poses a problem when you're reading papers or even textbook chapters, is that it may refer to Nectria and you know about Fusarium, but have not committed to memory that they're the same thing. Okay, so be aware of that. Sometimes there'll be two names for the same fungus. I'm really sorry. You just got to pay your taxes. That's something we're dealing with as mycologists. Okay.
Is there any resource where we can look up the name and see if there's another name for fungus?
Yeah, and I'll show that to you when it becomes more important. Yep. If there's like a synonym lookup. It's really depressing when you go there because there'll be like 8 different synonyms for the same species. And it gets really confusing. I am of the opinion that species aren't even a real thing, and so I tend to not pay too much attention to it. So I don't expect you in this class to memorize any of the dual names for any of these species. I'm more concerned in this class about you learning the major groups and like their life cycles and that kind of thing. All right. So here's a few phylogenies essentially showing the same stuff. These are the major groups. And so these are the ones we're going to be going through. Okay. The chytrids, that's what we'll start out with. Then the zygomycetes, which aren't actually a real group. And then the glomeromycetes, which probably aren't a real group either. And then the ascomycetes and basidiomycetes, which are definitely real groups. Right? So these are the main groups. The earliest of these is the chytrids. And by earliest, I mean they most closely still resemble the common ancestor of all fungi. Okay. So here's chytrids. When you pick up your lab packet from the office, I'm going to give you an easy way to find these. They love to grow on pine pollen, all right? And so I've got a little 1.5 mil tube with a bit of pine pollen in it. You're just going to stick some dirty water in there from whatever random pool, non-chlorinated, right? So next to your house, if there's some stagnant water, if you go out to a creek or a lake or something, put some water in there. Chytrids will show up and you can see them on your microscope. So this is a chytrid. It literally means little pot. Okay? And that's because their main body form is basically a little sphere. Okay? And they've got these hyphae that come out and kind of move into the pine pollen or the amphibian or whatever it is they happen to be eating. Okay? Here's some on some algae. Close up of them on some algae, and you can kind of see in this third picture right here, the scanning electron micrograph. You've got an algal strand, and then in the middle of it, you can actually see this like hyphal structure. That's the chytrid filament growing through the algae. Okay, so these are chytrids. This is their flagellated state. We call that a zoospore, okay, because it moves around like an animal, just like zoology. Okay, so this is a zoospore. This is a zoosporangium. What's that mean? Who remembers from 1620 megasporangium, microsporangium? Uh-oh.
I remember.
That doesn't
Is it
bode
like,
well.
was it like the spore producing organ, basically?
Yeah, that's right. Spore producing organ. A sporangium is a structure that houses spores. Okay. A zoosporangium is something that houses zoospores. I know the terminology is new, but you're gonna start seeing patterns. Okay. So get used to sporangium. All right. So this is a chytrid here. If there's its zoosporangium, it'll actually burst open. And so here's an image showing it bursting open. You can see all the zoospores moving around in the zoosporangium. And then once it bursts open, they're going to just kind of be set loose. And they swim around in their environment, going crazy. Okay. Those zoospores go out, they find a new substrate, they land on it and grow a new chytrid body. All right. So that's zoospores being released from a zoosporangium. We define chytrids not very rationally, just everything that has zoospores, we call it a chytrid. Okay. Here's the life cycle of a chytrid. Okay. We're going to start with our zoospore right up here in the top. All right. Those are haploid. So this is basic life cycle. If you remember anything about plant life cycles, this should be pretty straightforward. Haploid zoospore germinates. It grows into its substrate and starts building a gametophyte. What do gametophytes make?
Gametes.
Gametes. Yep. Okay. So here's the mature gametophyte. It almost looks like an alga or a plant or something, right? It's got male gametangia and female gametangia. Hey, we're using botanical terminology for some reason. Yep, sure are. So it shouldn't be too hard. They make male and female gametes that look exactly the same. They're both little like zoospores, except one is bigger. Which one is bigger?
The female.
Yeah, the female's bigger. That's how we define female in biology. It's the bigger gamete. Okay. So they get together, they meet up, and now they undergo karyogamy. So they're fertilized, and we've got a diploid organism. Call that the sporophyte because it makes spores. Okay, so here's now an alternation of generations. We've got a multicellular organism that's diploid. Okay, it can make diploid zoospores. They can go land somewhere, create a new sporophyte, which can then make diploid zoospores, which make a new sporophyte. When things go south in the environment, temperature change, running out of nutrients, something like that, that's when we undergo meiosis. Okay? If things are good for you, you don't need to undergo genetic recombination because clones of yourself are going to be doing just as well as you are. If things are looking bad for you, it's adaptive evolutionarily to mix things up, randomly roll the dice, and hope that those offspring are going to do better in this environment. And so it undergoes meiosis, and that's where we get our haploid zoospores, making our multicellular haploid gametophyte generation. This is just like the alternation generation for plants. Okay. So that was the most complicated chytrid out there, that Allomyces. Okay. More often, chytrids are a bit simpler. They're this holocarpic type where it's just like a little ball. Makes some spaghetti noodles that go in and eat a substrate. When it runs out of juice, it makes zoospores that go out into the environment. Okay. But you can see there's a lot of different growth forms for chytrids, and that's possibly because they're not actually a monophyletic group. They're just things with zoospores that we don't know how to classify properly. Okay. If we were better at growing them, we might have gotten further in their phylogeny, but they're pretty hard to grow. Here's some actual photographs of chytrids. These are what you're going to be finding here in the top left when you add some pond water to your pine pollen. The Mickey Mouse shape here is a grain of pine pollen. The circle is a chytrid growing on it. Okay, it's one of their favorite foods. So they tend to live in aquatic habitats and wait for things to come along that they can eat. Here's one eating some algae, living algae. Here's one eating something or other. And then, of course, chytrids are recently famous for basically making all of our amphibians go extinct all over the world. Okay. We're going to read an interesting paper about how this happened, where they came from, and how it relates to climate change. But essentially, chytrids started here in Central and Northern Southern America and have spread globally in the past like 50 years, wiping out entire amphibian populations, a lot of local extinctions all over the place, because they live on amphibian skin. They're not eating the frog, they're eating those secretions that it's making, for instance, or digging into some keratin being produced. Okay, but that clogs up the frog's skin. They use their skin to breathe, they suffocate, and they die. Right? So chytrids are ecologically important for that reason, especially recently. Okay, so they may seem tiny and insignificant, eating some pine pollen, but they are everywhere. We find chytrids even in Antarctica. Okay. We got a lot of ecological modes. A saprotroph eats dead stuff. That's your decomposer. Okay. A biotroph requires living tissue to parasitize it. Okay. And then a necrotroph does both. It needs living tissue, but only so it can kill it and then eat it. All right. So that's the sociopath of the fungal world, it's the necrotroph. Okay. In chytrids, we even have some obligate anaerobes. Who wants to pronounce that word for me? Somebody, give it a shot. We all want to laugh at you. Anyone?
Neocali masticates, or gales.
Yeah, good job. Okay, you kind of deprived us of a good mocking opportunity, but that was it. Yeah, Neocalamastigales. So fun word to say. And then these are in the group Neocalamastigomycetes. These are the gut fungi that live in cows and do the digesting of grass for them. Okay. So when you read in chapter 6 of Webster and Weber, you're going to see all these different biochemical groups, like nice examples from the chytrids. It doesn't really go into the Neocalamastigales because they're relatively recently discovered. They do have zoospore stages, but they're quite different from the rest of chytrids. But ecologically and agriculturally very important. I don't know if you know this, but cows can't actually digest cellulose, like at all, but they live on grass, which is made of cellulose. Right, so it's bacteria and fungi that are actually breaking it down for them. Same for termites. The Neocalamastigales live inside of termite guts, and that's how termites can eat wood. So they're actually feeding this fungus wood, it's fermenting it, and they get to live off the byproducts of that. Okay, so Neocalamastics, this is what it looks like. What's weird about it, and why some people are kind of freaked out that it's fitting into chytrids phylogenetically, like genetically, is that it doesn't have one flagellum. It's got like 6 to 8 flagella. All right. Things get weird with the chytrids. Okay. So, towards the end of each batch of lesson slides, I've got a slide like this that kind of just has questions for you. these are the sorts of things I want you to consider. How do these fit into our picture of fungal evolution, morphology, physiology, diversity, and ecology? Okay, so what is it that you know about chytrids, especially after reading your chapters and the paper, that fits into what we think about as being fungal? How does it change your definition of what a fungus is? Or how does it fit neatly into your definition of fungus? Okay, I filled out the first one for you. In subsequent lessons, that's for up to you to fill in. Okay, so in this case, how do they fit into our evolutionary definition of fungus? Well, these are polyphyletic, meaning we're not really clear on which groups are sister to each other. They seem to be only defined by the fact that they have zoospores, okay? But they're really different from the rest of fungi. Okay, so these are early diverging. That James et al. 2006 paper that you're going to read really covers this nicely, and they give you a really good overview of the evolution of flagella, or where it was lost and reacquired. Okay. Morphology, they are hyphal, so we know that hyphae evolved really early in fungal evolution. They've got flagellated spores, so just like animals. Okay, so that fits in with what we know about what's similar between animals and fungi because of common ancestry. They can do sexual and asexual reproduction, like the rest of what we'll see in fungi. Okay. They've got simpler hyphae than some more quote-unquote modern groups of fungi, but they do have hyphae. They've got complex secondary compounds and excretory proteins. Okay, so you can see how I'm just trying to go through and update our definition of what it means to be a fungus based on what we learned about chytrids. And then the last slide is going to be your assignments. So just like on the website, all right? So you know what? I deleted that requirement. Don't do number 4. Everyone cool on that? I'm going to write it down for you in the chat. Don't buy Mr. Bloomfield's orchard. I was going to have us read that because it's kind of a pop science book about fungi, and I was going to have us read it in the second half of the class, but I figured you had enough reading already. Okay. It's a good book if you're really into fungi and want something that's easy to read, but we won't do it required. Okay. So there's your assignments. I'm going to exit out of this. Come back up here. Okay. When is this class over?
Good question. I think it's 12:40.
Yeah, we got all the time.
Is that right?
Yeah, 12:40.
Okay, so we got a bit of time. So let's chat about fungi and do questions and answers. Anything is game. I'll stop sharing my screen. All right. So anything about the course structure, questions about chytrids, about the reading?
Yeah.
Microscope.
When are you going to be in your office
15
for us to pick those like packets up?
The packets, I've got my lab assistant making your agar dishes this week, and so I hope to have those ready to pick up next Monday. Did that answer your question? You were cutting out a bit there.
Yeah, thanks.
Okay. Yeah, and I'm just going to stick those in the main biology office. You can come check your name off the list and take your packet. It's a tube of 20 Petri dishes with agar, a little instruction card, some already growing fungi that we're going to do like genetics experiment with, and then some just supplies for working with your tiny microscope. If you buy this microscope, it comes with a slide, and it's the worst slide I've ever seen in my life. It's like this plastic that's hard to see through. scratches, garbage. So I'm giving you some slides and cover slips also. I've got videos on the website of how to use these things along with pictures from it. It's kind of a neat, if not super clever, somewhat functional clip. And you put your phone camera right over the hole and it clips on. So you can take photos of the stuff you're looking at. And the clip comes off, you just want to look through it. So it's kind of neat. I've tried a few of these pocket microscope things, and this one actually works. You can see red blood cells with it. Tested that out, so it's pretty cool. Other questions?
You said we pick those things up by just coming just to your office, right?
No, just the main biology office. So just when you walk into SB 243, like the front desk, I'll have them there for you.
SB 243, you said? Okay.
Mm-hmm.
And will we be able to pick those up basically any time of the day as long as it would be a normal work day?
Yeah. Mm-hmm.
Okay.
Any questions about expectations for this class? Fungal terminology?
About the tests, did you say they're open book?
Yeah.
Okay.
You can sit and watch the lecture rerun while you're taking the test for all I care. It doesn't matter to me. I'm not going to make them like extra hard because they're open book either. It's for me, it's not whether you've memorized the names of fungal teleomorphs, right? Or if you know that Mycota refers to a phylum. It's that you're familiar with the terms. You've gotten over the learning curve kind of hump for mycology, and that you can look stuff up efficiently, right? I don't know any professional mycologists. I am one myself, and I've got a lot of friends who are, that have all that stuff memorized either. We just have been around it long enough that we are good at Googling fungal stuff. And so that's where I'm trying to get you to be, is kind of good at looking this stuff up. I mean, obviously a familiarity with being able to decipher that a zoosporangium houses zoospores, right? That's going to be pretty handy as you take an exam, but it's something you can look up if you're desperate. Yeah.
Okay.
We appreciate that.
Thank you.
Yeah. I don't want to turn mycology into like a chore for you, because it's freaking awesome. Like, fungi do amazing stuff. They're fun to look at. They grow in your bathroom and on sandwiches, and they used to be the biggest organisms on Earth, and they're still super important for all kinds of stuff. It's the reason you get to have hamburgers now, is because cows have Neocalamalagastes fungi in there, and that's all fine and well, but if I turn it into a chore, you're going to hate it, okay? I don't want it to be that. I am going to make you do that reading, because that's how you learn about this stuff. Yeah.
Right.
But don't do the reading to memorize.
Okay. Do the reading because it's interesting and we need to learn it still.
Yeah, it is interesting. I think if you approach it that way, you'll actually enjoy it. If you're reading it to memorize and like highlighting and taking notes and stuff, that takes all the fun out. I don't like that. That's not how I learned mycology. So
okay,
I'm going to spare you that.
Thank you.
Yeah.
Will the test be timed?
24 hours. Yep. So technically, yes.
Do you base your exams just off of like slides and reading, or what do you?
Yeah, slides and reading, and also perhaps like some of your results from the labs that we do. So I may have like an exam question that's like, post a picture of the bread mold that you grew, right? Yeah.
I saw on your website, you mentioned that you like to have discussions in Slack.
Yeah, we can do that here if you prefer.
Oh, I'm just not sure what that is.
Yeah, so I had that set up before the university told me I had to use Microsoft Teams for everything.
Oh,
So it's an alternative to Teams that's not so Microsoft based, so it's a little like more accessible. I run a Linux computer, and some people are on Mac, and not all the Teams features are available on Mac. So I was going to do that, but for just chatting and stuff, Microsoft Teams is working pretty well. I'm pretty happy with it so far, so that's what we're going to stick with
okay.
by default. Yeah. So yeah, on Thursdays we're going to meet here just as before. I'm going to hit record, and we're going to chat about the reading, that kind of thing, talk about upcoming assignments and labs. It'll just be here. Yeah.
Awesome. Thank you.
Mm-hmm. All right, well, I guess if you want to leave, you're welcome. I'll stick around until class is actually over for people who do have questions or want to chat about something. Make sure you're using this website. It took me a buttload of time to put together, so don't waste it. All right. It's got all the stuff on there. It'll help you keep on track this semester. And things are weird right now. I don't know if you noticed. But like we're all sitting around in pajamas and we're in class. It's kind of cool, honestly. I got my dog with me. This is awesome. But it does pose some challenges, and key among those is motivation and engagement. Like, there's definitely an incentive for some people to maybe mute their microphone and video and walk away for 45 minutes and come back at the end. I can't stop you from doing that, but it's not going to work out for you. And so I'm hoping that we can try to be as engaged as possible, especially on Thursdays. You're going to be required to chat if you want points on Thursdays. So be ready for that. Just try to stay motivated. This website should help keep you on track because time doesn't exist as normal right now, you know? Time's a flat circle. So let's try to stay focused, be task oriented. You don't have to put on real pants. You can wear pajamas. I don't care. That's fine. But stay on track with our week-to-week meeting schedule and your tasks. All right.
I got a question.
Yeah.
So let's say I did want to do some DNA analysis. Is there somebody at the university I could go talk to get that going?
Yeah. I mean, well, the trick is that on-campus research is pretty limited right now this semester. Yeah. But there are plenty of people in the biology department who could help out with that. Yeah. And normally I'm doing it every week in my lab. Not right now. There's a chance, and you can shoot me an email if you want more details about who to contact or whatever, but your options are somewhat limited, but the long answer is that yes, there are 28 faculty that are doing DNA research on campus under normal conditions. Yeah. Was there something in particular you had in mind?
Well, I mean, I'm just pretty interested in genetics. That's what I hopefully want to go to my master's for, but I don't have a whole lot of experience in it because there's a limited number of courses that offer it, and especially a limited number those past couple semesters. So
yeah,
I kind of just want to get my feet wet and get into the environment where I can at least see it happening, you know, hands-on sort of thing.
Gotcha. Yeah. And I apologize. I mean, obviously the pandemic's not my fault, but I'm still sorry that we don't actually get to do that because that was going to be a component of this course. You're not missing out on a whole lot. I'll be frank with you. It's super boring. You're moving small, like infinitesimally tiny bits of clear liquid from one tube to another, and that's what the genetics lab is all about. To me, it gets exciting when you get your data back from the sequencer. Like, That's when you got something real and tangible. I've spent hundreds and hundreds of hours of my life getting carpal tunnel syndrome from pipetting back and forth between tubes, and I've never enjoyed a second of it. I don't know many people who do either, and those jobs don't pay very much because robots are increasingly doing that part of the genetics process. So what you will get in this class is a chance to play with DNA sequences. All right, so pretend that the robot just did all your work for you while you relaxed and had a cup of tea, and now you've got your actual data. So we're going to get to at least do that part of it. In future semesters, I don't think it'd be too hard for you to get the horrifying experience of pipetting and doing PCR. So we can chat about that.
Okay.
Don't lose sleep over it. You're not missing much.
All right. I'm not gonna get rejected from a graduate program because I haven't ever pipetted.
No, you're more likely to get rejected from a graduate program because you've never done bioinformatics.
Which I'm in your class, so.
Great.
Dr. Zahn, will you show us your dog?
Yes.
All right. Andy.
What's his name or her name?
His name is Indiana Jones.
Oh, Indy. Okay.
Here, buddy. No, come on up.
Oh, so cute.
He's an old man.
Yeah, he is. Oh, how old is he? He looks like 13, 12.
He's 9, but he's got like
Oh,
purebred boxer problems. He's a rescue.
oh, I'm sorry.
He's good.
Such a cutie.
All right. Oh. Good boy.
Thank you.
Yeah, he's got to sleep in here with me while I do class.
I'm sure he loves it.
All right. Anything else?
I have a question, but I don't know if it's applicable to anyone else, but I just got back from Romania on Friday, and so technically I'm supposed to be quarantined and not allowed to come to campus for 2 weeks.
Is there somebody you could send?
Yeah, that's what I was gonna ask. So I can just have someone come and pick it up.
Yeah, absolutely.
Okay, perfect. Okay.
There's not going to be a security guard.
I didn't know if they were gonna ask for like our ID or something. I don't know.
No. In fact, I made a couple extra kits, assuming that some idiot student's going to walk by and just steal one. So it's okay. Yeah,
Okay.
they're just going to be out in a box.
Okay, great. Awesome. I can definitely ask someone to pick it up. Perfect.
Cool.
Thank you.
Mm-hmm. Well, I think we're all going to survive this semester. I'll do my part to get us through with a minimal of pain, as long as all of you kind of keep showing up and doing your reading. All right.
Will do.
So go read.
All right.
Thank you, Dr. Zahn. Appreciate it.
See you guys.
Thank you.